Yeah, it's in India. Which part of India?
Oh.
Oh, okay. This is from Punjab, India, Ludhiana. This is one of the Borlaug Institute South Asia sites. So most of our UAV pictures are just like pointing straight down, so this is one of the more interesting pictures from UAV. So I'm going to talk about how we can think about using secondary traits. So you go back to the classic literature, Falconer, and you look at like indirect selection. And so with high throughput phenotyping, we can enter into a new era of this idea of secondary traits and indirect selection. So that's what I'm going to focus on today. So this is just a snapshot. This is the world of wheat. And it's just highlighted by production intensity. So we're I'm at Kansas State University, which is right here in the center of this dark area of the Central Plains, huge wheat production region. We work a lot in South Asia. We've got field trials across North India and Pakistan. Be some of the data I shared today. And so this gives you an idea. Dr. Norman Borlaug is a famous wheat breeder, and so give you this idea that to advance both society and food security, we want to be focused on increasing and stabilizing increasing production. So the context of wheat is it's a staple crop, one of the big three, about 60% projected increase in demand. Okay, and then 20%. This is mostly due if wheat is pretty lousy under high temperatures, and so if you look at most of that wheat growing area of the world, it's already above the optimal growing temperatures for wheat. So heat stress is a big target for wheat. So that's about a 2% per gain year. If you look at most of the current studies that are estimating the rate of gain in wheat, it's about 1%. So we need to throw everything we can together to try and increase the rate of gain. And so this is what that looked like projected out here. We push this up with agronomy and breeding, and then we have a whole number of factors that are continually pushing this down. So it's trying to fight those off and hit this projected gain that's needed for the next few decades out to 2050. And so to put that in the context of the breeding cycle, probably very familiar. In wheat, what this looks like is you have crossing, you're making two-way crosses or top crosses, and then you have this really long evaluation period. And so early generations are selecting for diseases and disease resistance, primarily some agronomics and maturity. And then you go to yield testing, and then final stages, you actually do quality testing on wheat. And so obviously the population sizes get much smaller as you go through these stages of testing and selection, and the phenotyping costs go way up. So genomic selection, for example, is a really prime target for yield and especially for quality. I'm not going to talk about that today, but that's been a lot of our focus. And so we want to think about what are all the different ways that we can optimize this selection cycle and increase genetic gain. And so when we put that into numbers, it's the classic equation just reformulated from Falconer and McKay, and this is a function of the selection intensity. And so this is one thing we think about when we look at high throughput phenotyping. The selection accuracy is another big thing that we look at for high throughput phenotyping. Not going to talk about, but genetic variance is obviously an important one. You want to decrease the bad variants but maintain the good alleles. And then years per cycle. So we want to decrease the number of years it takes to select the new parents. And so this is one thing we're thinking about, obviously with genomic selection, but you think about how you can do phenotyping in earlier generations and decrease the years per cycle. And so one way to speed that up is a genomic prediction. So I'm going to talk primarily about genomic prediction and then incorporating in these early generations, incorporating secondary traits for selection. And so in a really simplified typical breeding program, most of the wheat programs now are using some form of a selected bulk or a modified bulk selection. And that's to go through the first generations of inbreeding until you have fixed lines. And so you have parents who go through several generations of inbreeding, and then you'll derive from a single plant, you'll derive like either a head row, a single row, or a small unreplicated plot. And so in the CIMMYT program, this goes in shuttle selection, or in a winter wheat program like in Kansas, this is actually 1 year for each one of these generations. The big point is that here, though, is that we have this small plot test. And so if you work on wheat, soybean is another really good example. The people that work on maize don't get it because they can get enough seed from one plant to go directly to replicated testing. But there's a lot of crops here that the seed increases is not fast enough to go directly to replicated testing. And so you have this generation where the only seed you have is to plant like a small, less than 1 meter square area. Okay, so this is the stage in the breeding program focus on today. And so in most of the programs, we're also genotyping the entire set of materials that come through in this stage. To give you an idea on numbers, going from this generation to this generation, in the CIMMYT program, it's about 50,000, and then we'll select out 10,000 fixed lines that go to replicated testing. In the Kansas program, it's about 3,000 that go to about 500. And so give you an idea of the numbers. So at this generation, we're talking in the tens of thousands of breeding lines each year. So here's the way we kind of picturize a little cartoon to think about different levels of prediction models. And so in sort of the plain old simple implementation of genomic prediction, right, you'd have your genotypes here, and this big phenotype is yield. Right? And so you just directly predict these. And so you can think about a lot of secondary traits that we know are yield components, anything from maturity to, in wheat, we would talk about like the number of spikes, the number of tillers. You talk about seed size. Any of these traits are obvious secondary traits, or even disease resistance can be in here. Secondary traits that are correlated to yield and therefore have some predictive ability. So think about any trait that's correlated to yield would have a predictive ability to predict yield. And so what we can start to think about now is you have a genotype that actually makes up all of these secondary traits that make up yield. So from a and so this is kind of the biology of it, but in a statistical modeling framework, we can think about modeling these secondary traits either as direct predictors of yield. You can model them as correlated response variables to yield and actually predict from the genotype using these as correlated phenotypes. And this is just two different ways of looking at that model. And so these are a whole different suite of models. And the basic, when going through this talk, the big difference is we talk about this as like the univariate prediction, meaning that we're just singly predicting yield. And then a multivariate prediction is when we're predicting all of these secondary traits at the same time and using the covariance to increase the yield prediction. Okay, so that's the general scope. So when we think about this in a high throughput phenotyping, I think about like these different component traits that we can start to measure in high throughput fashion. So we're mostly focused now on the UAVs, using these unmanned aerial vehicles or the drones for phenotyping. And so we can think about whole suites of different traits that we're working to individually measure these traits and then put them all together. And so some of these are real direct, canopy reflectance. You can measure with thermal cameras. You can measure canopy temperature. You can measure things like ground cover and biomass. And obviously, as I list these off, you can recognize that all of them are correlated to yield in some way or another. So I'm going to highlight just a few of these going now on like how we're measuring them and then how they're predictive of yield. So really the first and most straightforward one is just canopy reflectance, and so this is a direct sensor measurement here. And so we do this with near-infrared cameras from the UAV, and you can use these vegetation indices that are basically the ratio of green and near-infrared to red light. And so for a dead leaf, you're absorbing all that near infrared, and for a healthy leaf, you're absorbing the red and reflecting the green and near infrared. Okay, so that's why we can use these as a good proxy for plant health, which then is obviously correlated to yield. And so this is what it looks like. This is one of our trials in South Asia. This is just a false color image of what that normalized difference vegetation index would look like. So we've been doing this for several years, deploying these UAVs across all of the breeding trials, measuring primarily multispectral cameras, and then and trying some new hyperspectral and thermal imaging for the last year or so. So the first one I highlight here is like measurement of plant height, and then how we can measure lodging. And so this is obviously an important yield component. So we can do this using like a digital elevation model. And so just like you can see in stereo, because you have two eyes, you can see a distance. So with the UAV, as it moves across the field, you see from different angles. And so with this, we have depth image. We can stitch all of the images together and get a depth map of the field. And so this is just an idea that most of the fields have, for any single point in the field, you'd have 10 or more images that cover that single point. And so what this looks like then is you can build a 3D point cloud and actually model the field in a 3D space. And so this is an example of one of our fields in Kansas. This is just a ground control point. It's a fixed point for our surveys. But you can see this is early season wheat. These are about 10 to 20 centimeters tall. And so you can see the 3D model here. You can see some of the tire tracks through the field. And so we can take the sum or the average of these elevation points and actually measure the height of each one of these plants, each one of these field plots as they grow. So here we can very rapidly then have plant height measurements throughout the season, so we can start to build growth curve models. We can also measure, obviously, final plant height. And so, then the example then is if we can measure plant height, we can obviously measure like changes in plant height, which would be lodging, right? So if you have a plant and then it falls over, you're going to have a change in plant height. And so lodging is obviously an important one. It has a negative impact on yield and also quality as the plants fall down and get dirty and wet. And so it can be time consuming if you get these huge nurseries. Like here again, I said, we're doing this on smaller sets now, but we want to extend it to the entire set of these early generation ones. So here's just an aerial image. You can see some of these plots are still standing well, other ones have fallen over. So the question is, how can we measure this with a high throughput phenotyper? And so what we have is this UAV phenotyping. We build these digital elevation models. And so I just want to highlight, this is work of Daljit Singh. He's one of the graduate students in my group, and did this work at the field sites in India over the last couple of years. And so just an example of what this is we have a flight that happens before a lodging event. So these lodging events usually happen when the soil is rather wet, and you get a fair wind that comes through, and you'll have a lot of lodging overnight. And then we have a pre-lodging and a post-lodging flight in each of 2 years here. And this is in Ludhiana. It's a location that's really prone to getting a lot of plant lodging. So what we also have is like a ground truth, or just a visual assessment of the lodging. This is just visually going through and scoring the plots. On an incidence, this would be 0 to 100%, like how much of the plot has fallen over, and then a severity would be how far over it's fallen. And then we can have UAV-based measurements of these. We can have the differential mean, just the average of how far it changes, and then we have a mixture model that measures both the amount and the proportion at the same time. So what this looks like then is we can have a pre-lodging, this is a digital elevation model, and so this is just a false color image on the elevation. And so you can see the red here is low values that would be like the soil, and then you can see like an irrigation channel right here, and then each one of these would be an individual field plot. And so you can see some of these, like the dark blue would be obviously plant plants that are taller. Some of these light-colored ones would be just overall genetically shorter plants. And so we can look at this post-lodging. You can see overall the elevations are much lower. Some of these are really low and pretty much on the ground. And so we can take the difference between these two time points and get a differential change. And so basically this is the change in elevation before and after lodging. So we can do that. We can for each one of the points within that plot, we can fit a mixture model. And so we can model the distribution of changes within that plot. And so here's just an example of, you can think about each one of these being a pixel in that image. That here we can so anything that's at 0 means that before and after lodging you didn't have any change in elevation. Any of these negative numbers would be the amount of elevation change from standing up to falling over. So here's an example where you had some proportion, about 17% of the plot was still standing, and then the rest of it was lodged. And this would be the amount of lodging. And then this would be a plot where basically the entire thing has fallen over. So we can take that, we can find that these are down here in this corner, these are correlations between the digital measures here, and then these are the visual measurements. And so you can see that the incidence and the severity are well correlated. So if most of the plot falls over, it falls over clear to the ground. And you can also see here, these are negative just because we're measuring them on inverse scales. But the lodging mean and the mixture model from the UAV measurements are really well correlated here and here with the visual measurements. And so we can, from the UAV, we can get a really accurate assessment of the amount of lodging based on the elevation models from the UAV. And then for each of these are all measuring at the same time point, but that had a pretty strong correlation to yield of 0.2. And so here again, when I go back to this is one of the component traits now that we can put into an overall model that is predicting yield. So when I go back to this here again, right? So we were talking about measuring plant height and measuring lodging. That's just an example of how we're measuring these. So now I'm going to shift and talk more about just using the vegetation indices directly and using those as predictors of yield. And so this is an example of doing this in early generation materials at CIMMYT. And so this is to give you an idea. If you remember back to that breeding flow diagram, this would be these first generation small plots. And so there are 2 rows, they're about 1 meter in length. Here's like an aerial image. These are actual sizes here, or actual relative sizes here. And so this would be an example of one of these small unreplicated plots. And so they're less than 1 meter square in square meter total size. And so, for example, in 2017, Obregon cycle, there was around 38,000 of these. And so you can imagine that it takes actually days just to walk through them one time. Grain yield measurements are not really reliable because they're small plots, and it's obviously also not really tractable to take those kind of measurements. So the only selection is just a visual selection. They're rated as early, medium, or late maturity, and then Ravi Singh, the breeder, walks through them very quickly and spray paints the ones that he wants to keep. And so that's so the selection criteria is looking at it for about a split second. You look at the plot and say, is it right maturity? Do I like the height? Is the disease package good? And you spray paint it or not. Okay, so obviously the heritability and the selection accuracy is good, but it's not what we hope it would be. Okay, those then advance to a replicated field plot, and these are planted on beds, and so that's why there looks like there's actually irrigation channels right down the middle of each plot. But a replicated field plot would be this size right here. So this would be 1 genetic entry in the replicated field test, and then obviously you'd have another whole replication of that field plot. And so following this selection, there's between 30 to 50,000 of these every year. For the next generation, they're selected down to about 9,000 that go into a 2 replication yield test. And so so now we want to look and see how well can we predict yield from this little plot into what it's going to perform the next year in a replicated yield test. And so this is some nice work. This is another graduate student, Margaret Krause, who's at Cornell, and she lives and works at Summit most of the time. So what this experiment looks like is that in the breeding program, each year, so say in the first year of field testing, you have these small unreplicated plots. They're visually selected, for example, these ones that are circled in red are visually selected, and then in the next year, they're advanced to these replicated field plots. And so for this, what we did is these are the first year PCs, meaning like small plots, and these are the second year and the first year of replicated yield testing. So that's what this YT is. We have these like other Margaret plots that we call them, for lack of a better name, that this is Margaret's experiment. And so we actually took a set of these replicated entries, right? So remember, each year in the breeding program, there's new sets of material coming through. So this would be across years. This would be within 1 year. We took a subset of these replicated lines and actually grew them in parallel in small unreplicated plots, mirroring what happens in the previous year. So this is within 1 year, and this would be across. This would be from here to here would be across years. Also applying selection between those two stages. So what this looks like is that we measure all of these with the UAV. And so on these sets, we have like imaging across all of the large plots. We have imaging across all of the small plots. And then on the large plots here, they would obviously be combine harvested, and you'd have pretty accurate yield data on them. And so what we have then is that on the small plots, we just have imaging, right? Because in the real breeding program, they would not be harvested for yield, and only the selected ones would be harvesting. So from this, we have a training set where we use 80% of the lines from the large replicated plots to build a prediction model based on the vegetation, on the UAV imaging. Okay, so then the target trait is obviously replicated plot yield from the large plots. And then the secondary trait is this normalized difference vegetation index. The remaining 20% are left here as a cross-fold validation. Okay, so this would be the training phase where we build the model here, then we basically use the UAV phenotyping from the small plots. And then the testing stage would be we take this set that was left out, image them in the small plots, use that to predict the large plot yield. Okay, so ultimately what we're doing here is taking spectral reflectance data from the small plots and predicting replicated large plot yield. Okay, so, and I should say too, we genotype these whole things so we can make genomic predictions on them at the same time. Okay, so here's the basic summary of that. We've done this across 2 breeding cycles now, so this is just representing yield data that came from the 2017 cycle and then from the 2018 cycle. And so remember also, because of the flow of the breeding material, these are new sets of genotypes each year, and obviously new environments too. Okay, so here what we have is the relationship matrix. This is using genomic information, so the relationship matrix derived from genetic markers, and this would just be the pedigree-derived genetic markers. And then each one of these boxes here would be a univariate, so this would be, if you remember back to the cartoon, this would be just a straight genomic prediction model right here. This would be a straight prediction using the pedigree blood, okay? And then each one of these other colors are multivariate prediction models using NDVI, green NDVI, or red edge NDVI. So just 3 different flavors of vegetation index. So the real comparisons you want to look at are comparing this red box to the other group of 3. And so you can see, for examples, that usually what we have is in each of these cycles, depending on if we correct for days to heading or not. Obviously, later maturity tends to be higher yielding, but you have to continually select against just being later and later. Okay, so here's the realized gain. So this is the prediction accuracy here. This would be the correlation between the observed plot yield and the predicted plot yield in the large plots. And what you can see here is a genomic prediction model, when incorporating the secondary traits, is substantially higher. Even after correcting for days to heading, you can still get an increase of accuracy from about 0.2 to about 0.3 prediction accuracy. And so what we're actually doing here, and then this would be an example for using just straight pedigree. And so this would be a case where we could use the high throughput phenotyping data. We would not actually be genotyping any of the small plots in this example here. And so you could use the pedigree relationship matrix combined with the high throughput phenotype data to have this level of prediction accuracy. So here again, what's happening is we're from the small plot that's only like less than 1 meter square, we're using the UAV measurements to predict fully replicated yield testing into the next year. And so this is the same thing. The accuracies were slightly lower for the 17-18 cycle, but you can see here it does substantially better on the raw predictions, and then just slightly better or the same once it's corrected for days to heading. And so what this looks like then, this is the overall graph to summarize that. If we take all of the information from the NDVI from the small plots, so remember this is the first year of testing in small plots, you take the NDVI value here, you take the grain yield observed in the second year, and we can get a correlation here. This is about 0.37, I think was the final number here if we put the full model together. Okay, so with the 0.37 accuracy, you can use small plot information to predict grain yield. And so here's what it is when we put it into practice in the real world, is that a subset of these, we had almost 15,000 breeding lines that were in these small unreplicated plots in year one. So remember, in the flow of the breeding program, Ravi visually goes through and selects these. Okay, so these were about 3,000 of those were visually selected by the breeder. Okay, so there's already a selection pressure on these, and then they're shown as replicated trials in yield 2. So now we have yield data for this second year. So the question is, if we use this high throughput phenotyping to further select out of those 3,000, what would be the effect of the population? Okay, so now we can experimentally ask, just by using the yield data that's coming through the breeding program, how much better can we do than just visually selecting it? Okay, so that's what this is here. This is the mean grain yield of all of the selected lines. Okay, so remember this is 3,000 that was selected out of 15,000, visually selected. So the grain yield on those is right between 6.2, 6.3 tons per hectare. Okay. This would be the percent of additional lines called with NDVI, with using that first year yield prediction. And so the example would be, is this would be like an additional 20%. So I'm just going to focus here on, say, a 50% selection threshold. So this would be Ravi, the breeder, selected 3,000. If we additionally cut 50% more down to 1,500, that would be this 50% selection threshold. Okay, and so this would be the increase in the average grain yield. This is percentage change over here. So by selecting an additional 50% culling, you could hit another 2% gain in that first stage of selection in the breeding program. Okay, so that in itself, right? So here again, you have to be a breeder to appreciate that, right? But an additional 2% from one, and here again, remember, this is not a whole cycle of selection. This is one stage of selection during that whole cycle. An additional 2% gain in that one cycle of selection is really substantial. So this is just looking at what happens with genetic variance. Obviously, if we call out, if we remove an additional percentage of lines, you're going to start decreasing the remaining genetic variance. But here again, that's expected, just like you would for any selection experience. As you increase the selection intensity, you're going to decrease the remaining variance. And then you also reduce the number of families that remain in the breeding program. But here again, this is the exact same thing, that if we increase the selection threshold, increase the selection intensity, you'd expect to decrease the number of families. So as we go from 0 to 100% additional selection, you can see the number of families drops off. So if we're using that example of about 50% selection, you would have reduced about 1/3 of the remaining families in that stage of the breeding program. And the same thing happens with polymorphic SNP markers. So that's the example then of using these vegetation indexes to select on yield in an example of like small plot early generation selection. So you'll see here also I've got highlighted some things like plant maturity, heading date, other things like spike number or spike measurements. And so these are things that we can't directly measure from images. We started late, so how am I on time? Is it okay? I can have much time as I need, as I want. Huh?
5 minutes.
I have 5 minutes?
Yes. Okay, 10 minutes, okay. Yeah, 5 minutes is all.
Okay, you're going to lose some of your coffee break. Okay, so this question I'll have to go through really fast is like how to move beyond direct sensor measurements. Okay, so if we want to phenotype flower color, like we've been doing, this vegetation index is just a direct color panel of the thing. So if you want to do flower color, it's a simple filter. You just filter the number of red pixels versus yellow or versus any other color, and you say, ah, yes, this flower is red. So if you want to do flower type, it's actually a really complicated function where you have to take into account the shape and the angle of the flower, also the color, the textures, the size. And so then you could say this is a lily and this is a petunia. And so this is where we get into deep learning and thinking about how to do what the breeder can also do by visually assessing all of these complex phenotypes. And so this is a convolutional neural network that we're using now to directly phenotype. So if the input is sufficiently complicated, there's not a linear function. And so for the vegetation index, we just have a linear function. We want to take an image that's complex and map it to a linear combination of trait phenotypes. Okay, and so I'll highlight this is work from a grad student with our collaborators at George Washington University, Hong. So to build this neural network, we originally started out with this phenotyping tractor. We can drive through the field and collect these really high-resolution images. They have a GPS, so we know each field plot that the image came from. And we have some germplasm association panel. It's on and onless and different maturities, and we have a biparental population. So what we end up with is like hundreds of thousands of images that are tagged with a breeder score, so a visual score we can assign to every one of those images. Okay, so this is the labeled image set that we can use to train a network. And so what we do is we take those images, we do a weighted classification, so we can weight those for percent maturity, or a really simple one is just on versus onless wheat phenotype. And so an example of what these images look like is here you can see an onless phenotype without the ons here, and a typical on phenotype with the real long ons. So we have thousands and thousands of images, and they're labeled one or the other. Okay. And so we can have, same thing, just like we do in the genomic predictions, we do like a cross-validation. So we train the neural network on part of the image set, and then we validate it on a part of the images that it's never seen. And so this is what we did. We had 700 plots, we validated it on, so 600 were used for training, and then a subset was used for validation of the network. And so for scoring on and onless, that phenotype I just showed you, here's on an image set. So the observed phenotype, we had a lot that were on, and then a few that were onless. And so you can see that these here and these here were correctly classified between the observed and the predicted, and we had a few that were off. Remember also, because as we go through the field, you can image each individual field plot many, many times, right? So you have dozens or hundreds of images from each field plot. So when you take a consensus from all the images that come from an individual plot, so we had about 700 plots, the 673 were correctly predicted as on, 32 were correctly predicted as onless, and then we had 2 that were wrong here. But that turned out to be 2 replications of the same variety. It was like a mixed phenotype, and so there was like disagreement between the visual scoring and the neural network. And so this was like an intermediate phenotype that couldn't be accurately classified. So we can take this and extend this to like heading date. So here again, right, everybody knows that maturity and heading and flowering time is extremely important selection criteria for yield, but it's very poorly measured in the breeding programs, right? So the breeders just go through and say, too early, too late. But nobody's actually like taking like what day was the actual heading on. And so this is why we want to do this for high throughput phenotyping, so we can get more accurate measurements, both to put into the genomic prediction models and to use as these secondary traits. And so this is an example of what this looks like. Here's before heading. You can see here that the heads are just starting to emerge from the boot. And then this would be an example image of the heading, the heads that are fully extended. And so this would be, and we have these time series images across the field plots throughout the whole season. So here's an example of the image set. You'd have these labeled as like 90%, 10%, 0% heading. And so very similar, same thing, except we did this training and validation across years. And so we trained the model on a set of data from 1 year and predicted into the next year. And so we had training sets from 2016 and 17 on the association panel. And then we predicted this biparental population in the 2017 data set. And so what this looks like then is we need to take these time series predictions, and I need to back up here. And so this is the final, this is the training and the validation and the testing set. And so here you can see, this is just a heat map showing for each of the individual images how well, versus the observed label versus the predicted label, how well they were predicted. So there's a little bit of variance there, but you can see that there's a nice linear correlation, nice linear trend between what was observed and what was predicted on an image. And this is again here for 0 to 100% heading. So what we need to do is then change that from percentage heading to a heading date, and so we can apply this logistic regression function here. This is just a standard logistic regression. We fit the phi 1, this is the asymptote, we fit that at 100% because they end up at 100% heading. They all start at 0%. And then we find the date and when this 50% intersection occurs. And so this is for some visual measurements. What we can do is we can put the visual estimates and we can look at the neural network. So the CNN is the prediction. So each one of these little squares here would be a date that it was imaged and then the prediction from the neural network, the percent heading that was predicted. And so you can fit this logistic regression and you can find the heading date using the visual, using the neural network. And so here again, the neural network would be, when the full system is put together, would be high throughput imaging, followed by the neural network, and so you could do high throughput measurements of these time series heading dates. Okay, so we can do this across each one of the field plots. This is just be an example of 2 different field plots. We scored these all visually because this was the training set, and then you can put the neural network and find the heading date for each one of those. And so we can do this across all of the field plots. What this would be then would be that 50% intersection for a heading date for each one of about 800 field plots that were in this experiment. So each one of these circles here represents a field plot. This would be the day that it was scored visually, and this would be the day that it was assessed at heading, 50% heading for the neural network. And so you can see across the growing season, there's a really nice linear trend between the two. Most of these are within 1 or 2 days of assessing heading between the visual and the neural network. And so what we want to do then is we want to make this actually like high throughput here again. So going from the vehicle, going back to the UAVs. And so it takes about 5 seconds per plot to drive this phenotyping tractor through the field, which is like sort of fast, but when you try and do like 10,000 breeding plots, it's gonna like literally take like days again. And so, and actually Robbie the breeder is much faster than 5 seconds per plot. And so we want to shift this over to like UAV imaging. So to get the same high resolution imaging, but with the UAV we can do much faster. We can do less than 1 second per plot. And so if my video works, I can give you an idea. So this is like a little video to show you. What we do is we take, this is a pretty high-end UAV with a differential GPS on it, and we take a really high-resolution camera with a zoom lens, and so you can fly at about 4 meters. So we're flying at about 4 meters. It's actually zoomed in to an area of only 1.5 meters on the plot, okay? And we take this in video, this is 4K video, and so you can get extremely high-resolution imaging and actually go really fast. So let's see if this works. Okay, so this is the UAV kind of flying across here. This is what would be coming through the camera. And so you can see this video. This is about how, this is like real time right here. And you can see each one of the field plots as it goes by here. Okay. And so what we do is we can, to image the whole field, you can traverse up and down these ranges right here. So you'll fly down one range, turn around and fly back down the next range. And so with this, we can image this entire field. This experiment was about 1,000 plots, and it would take about 25 to 30 minutes to image the entire field at this level of resolution. And so now the trick is just to how to crop out, because we know the order that we fly through the field, we know if we can segment out the individual plots, you can tell which image video frame comes from which plot. So this was nice work that's done by postdoc Kevin in my group, and he basically just applied a bandpass filter and then these Gaussian kernels to get the frequency at which these plots pass by in the video frames. And from that, you can identify the single frame. Let me go here. From that, you can identify the single frame in the video that corresponds to the image that's directly in the middle of that field plot. Crop that out, and then we can get these thumbnails that have the same resolution as the phenotyping tractor. And so if you zoom in on one of those, this is an actual thumbnail from that video frame as it's flying through the field. And here, this is onless. You can see this phenotype nicely here. And here's a typical on phenotype. So we've taken this initially, put it through that same neural network, and we can hit that same level, 99% prediction accuracy for this trait. We're still working through the huge set of data for the time series, doing this time series for heading date, but it's coming. Okay, so that's where I just want to summarize, and here again, right, to say, like all of these individual component traits, I highlighted some of these, how we can predict yield really well already, and what we're working towards is incorporating all of these secondary traits that we can tractably measure with the UAV high throughput phenotype, and then subsequently we can build these into just a classical statistical framework for a prediction model, or we can also shift these into the crop growth models that have been coming out of like the Pioneer, I still call them the Pioneer group, Pioneer group with like Charlie Messina and these guys. So that's the summary. I showed you some of how these secondary traits can be modeled here, and I think the real interest in our group now is that as we can fill in this layer of phenotypes here, it opens up a really interesting world of like new types of prediction models and new ways that we can apply secondary trait selection in the breeding program. So that's everything. I got a really great group and a lot of great collaborators to acknowledge. And so I just want to highlight a couple of my group. Byron Evers is a technician who manages all of our field trials and also does some of the UAV flights. And Mark Lucas is, so for all your students, Mark is a database programmer in my group. And so, like just for example, like one day of UAV phenotyping, we pull in about 500 to 800 gigs of data. Okay, right? So we generated about 5 terabytes of data just from the UAV. That doesn't count like sequencing data and stuff. So having a database programmer is extremely important positions. Daljit, I highlighted some of his work on the lodging, and Kevin is a postdoc in computer science, postdoc in my group that's doing these really nice processing pipelines. Also highlight Rich Brown and Grant down here are UAV pilots in the group. All the collaborative work at CIMMIT, this is under direction of Dr. Ravi Singh, is a breeder. And the deep learning work is in Robert Pless's group. And I also highlighted some work from Margaret on the secondary trait prediction. And this is largely supported from National Science Foundation in the U.S. and a U.S. Agency for International Development. So I'm going to really stop there, taking questions.
Okay, thank you, Jess. Now we move into the discussion session, and I'd like to ask to anyone who has a question or comment to make to please raise your hand. Natalia?
Jesse, so in your correlated objects, what do you think that NDVI is adding in terms of, is it a physiological stamp? Is it environment? What is it?
Yeah, we don't really know. So it's something. So it is something that Robbie can see, and it's also something Robbie doesn't see or doesn't select on, or he's going too fast. So Robbie, to get through that many, Robbie goes through 3,000 or 4,000 in an afternoon, right? So literally, you have to walk pretty fast to keep up with him. So if I didn't show it, but if you look at the unselected and the selected, Robbie shifts the NDVI values also. So if you look at his unselected versus his selected set, they've shifted maturity and they've shifted NDVI values. So Robbie's selecting on that. It's probably a combination of, right, like just overall health, disease resistance, and then some level of balancing maturity and, like, you know, just properly translocating.
Okay.
So
So is this something that you think could help understand? Because you did this across environment. So is this something that could help you figure out what are the relative responses of, you know, 1 year and the other year, and then as they correlate to yield, or is that way too?
No, you could. So the question was, like, can you use this to understand and better predict G by E, right? If I rephrase it. So yes, I mean yes and no. So in this example, we're not trying to do that at all. All we're trying to do is, like, predict the yield of a genotype that's in a small plot if it was in a replicated yield plot in the same environment. But some of the other stuff we're doing is, like, trying to use this data to understand how to predict G by E. And so I didn't highlight it, but Daljit's also been doing some nice work on ground cover. And in some environments, it's really predictive of yield. In other environments, it's just not predictive at all. And it has to do with, like, thermal time. And so we can start to understand that, but yeah, that's a different question. That's more like predicting G by E once they enter the advanced testing.
Right. I mean, and using the phenotype.
Other question?
Great talk, Jesse. I'm trying to understand, like, the model that you're using in this using the NDVI to make the predictions, because what you described it as if you were training. There was a statistical model built.
Okay. Training is probably wrong, right? It's the training set versus the validation set. Is that what you're
I mean, ultimately, it is a single number, an NDVI, that's used to make a prediction?
We can treat it a lot of different ways. In this way, it's just a vegetation index. So it's a 0 to 1 number. Okay. It's a single number. So you have a single number and you have a single number for yield. Right? So you basically have two responses in this particular model.
But the prediction that you're making, the predictor variable is simply you don't have a yield number for the plots you're trying to predict. All you have is NDVI.
Yeah.
So what more can you be doing except selecting for high NDVI? I'm not sure how you can get anything more complicated happening besides simply selecting, like, for a high value of NDVI.
We have the genotype.
So if you don't have the genotype data in there.
Then it would just be a straight linear model of NDVI on the yield.
And you didn't really show that comparison. What does that look like if you don't use the pedigree or the G matrix for that analysis?
Oh yeah, that's a good question. We've looked at that a little bit, and sometimes it works great, and sometimes it totally collapses, and we don't know why.
Okay.
So the point is, it never works better than having the genotype or the pedigree in there. Sometimes it does just as well, and sometimes it doesn't predict at all.
Okay. Also, the other question was just logistically, how do you envision this being used? Because you're going to fly your drones and take some data, and you'll have a computer make some predictions about 20,000 small plots. that's too many. There's not time for Ravi to look at a PDA and look at that number from the prediction and integrate it with his breeder's eye and make a decision. He's going to have to walk it the way he always walks it, and then you're going to have to somehow either additionally call after based on your predictions. Is that what you're thinking, or?
Yeah, well, we're also working on like an augmented reality, so it will display it as he walks through it. Okay, but in the real world, right, it'll be a combination of you go through and you paint the ones that you want to keep, right? You predict those also, and preferably before harvest, you predict them and further call out, further reduce it by 50%. This set that actually gets harvested, right? Because once you go out there and you hand harvest these, that's when the labor and the cost really go out. So ideally, right, you'd go through and visually select them, you'd layer on top the predictions, and then you'd select a smaller subset that actually gets harvested. So yeah, we're close to that. We're still working on that augmented reality.
Okay, if you have more questions, you can follow up with Jess during the break or tonight during the dinner. So I'd like to thank you, Jess, on behalf of GBank and the organizing committee. I invite you